Welcome to PGE Pathshala, paper number 8, Biology of Parasitism, module 33, Plant Parasitic Nematodes. I am Dr. Vartika Mathur. You have already studied about nematodes in an earlier module in this paper. After completing this module, you will be able to understand the economic and ecological importance of plant parasitic nematodes. You will also be able to learn the phylogeny of the plant parasitic nematodes as well as understand their biodiversity. You will be able to identify and classify them based on their morphological features and mode of parasitism. You will also be able to distinguish them based on their trophical niches. Plant parasitic nematodes are usually small soil-borne pathogens, and the symptoms they cause are often non-specific. They are extremely diverse and abundant in all types of soil, and plants regularly encounter them during their lifetime. There are more than 4,100 described species of plant parasitic nematodes. With only a few exceptions, all these nematodes are root feeders. So what can these tiny organisms do to plants? Are they of any economic importance at all? Well, their presence is not only the key driving force of plant succession in natural environment, but they also collectively represent the significant constraint to the global food security, with annual crop damages estimated to be more than $80 billion per year. Nematode infections affect the plant at different levels. Infested fields show a reduced number of plants, and infected plants have a lower biomass due to a disturbed physiology. Crop damage is not only limited to the direct nematode-plant interaction itself, but very often nematode-infected plants are more susceptible to fungal or bacterial infections. Certain nematode species can serve as vector for a number of important plant viruses, or may enhance crop losses associated with insect as well as weed pests. Now let us move to the phylogeny and diversity of nematodes. Most of the nematode species are microscopic and lack obvious distinguishing characteristics. Therefore, classification within the phylum has been very difficult. However, based on small subunit ribosomal sequences of 53 species, Blackster et al. Proposed a molecular evolutionary framework in 1998 for the nematoda, comprising 5 major clades, all of which include parasitic species. In this picture, we can infer that animal parasitism arose independently at least 4 times and plant parasitism 3 times. Evolutionary adaptation to plant parasitism led to some striking morphological changes. Because of this, plant parasitic nematodes show huge biodiversity. Almost all plant parasitic nematodes possess a hollow, protusible hypodermic needle-like axial sphere which serve as the interface between plant and the nematode. Now, what are the adaptations to plant parasitism? The axial sphere serves 3 functions. It can be used to penetrate the plant cell wall. It can withdraw nutrients from the cytoplasm as well as release secretions. These secretions are synthesized in pharyngeal glands, which have enlarged due to adaptation considerably when nematodes are adapted to parasitism. 2 different types of pharyngeal glands are recognized: subventral glands and dorsal glands. Each gland is a single cell with a long cytoplasmic extension. It terminates in an ampulla. The ampulla then connects to the pharyngeal lumen via a valve-like structure. Secretory proteins are processed in rough endoplasmic reticulum. and Golgi apparatus. These are then collected in secretory granules. These granules are transported via microtubuli towards the ampulla. Here, secretions are released into the pharyngeal lumen. Let us now move to the biodiversity of plant parasitic nematodes. These nematodes occur in three widespread orders: Triploncida, Dorilemida, and Tylencida. The majority of these nematodes are in the order Tylencida. Although there are some similarities between these taxa, it is evident that evolution to plant parasitism occurred convergently in the three orders. Let us first see Triploncida. All triploncid plant parasitic nematodes are migratory ectoparasites on roots. The nematodes in this order possess 5 secretory glands, 1 dorsal, 2 anterior, and 2 posterior subventral glands, which end in the basal bulb of the pharynx. Their stylet, also called as onchiostyle, is a modified mural tooth. Trichodoridae, a family in Triploncida, explores the surface of the root and feeds on the epidermis cells. During feeding, which rarely exceeds a few minutes, the ventrally curved stylet is continuously thrusted into the perforated cells. Injection of secretions result in the formation of feeding tube. Due to the relatively large width of the feeding tube, they are the only root parasitic nematodes capable of removing large organelles from the cytoplasm. Now, as if this was not enough, and if epidermis cells survive the feeding, plants become prone to tuberovirus infection. Trichodorids are considered to be little advanced in their mode of parasitism, as the cytoplasm of several hundred cells has to be withdrawn to produce a few eggs within a week. This picture shows the schematic representation of feeding sites of some migratory root parasitic nematodes. In this, the first number indicates the feeding site of triploncid nematode Trichodorus species. Next, we move on to Dorilemida. The eggshell sphere of Longidoridae, which is a family in the order Dorilemida, is composed of odontostyle and odontophore. Odontostyle is thought to have evolved from a mural tooth. It is formed in the anterior part of the pharynx and moves forward during molting to replace the previous odontostyle. On the other hand, the odontophore is reformed in situ. The length of the stylet enables Longidoridae to feed deeper inside the root tissue. Nematodes in the order Dorilemidae have 3 pharyngeal glands, 1 dorsal and 2 subventral. Most of the time, root tips are selected as feeding sites, and root tip galls, Xiphinema index, another species in this order, feeds on a column of cells. It starts feeding for a few minutes on one cell and afterwards progresses to deeper laying cells. Dorsal gland secretions are injected into the cells and seem to liquefy the cytoplasm. After ingestion of solutes by the nematode, the cells die of necrosis. Probably also in response to secretions, the neighboring cells become hypertrophied and binucleate. In case of Longidorus, stylet secretions seem to induce a gall-like structure containing uninuclear hypertrophied cells surrounding the necrotic feeding cells. In this figure, the number 2 and 3 represent the feeding sites of dorilemid nematodes. The second one shows the feeding site of Longidorus elongatus, and the third one represents the dorilemid nematode Xiphenema index. Lastly, we move on to the order Tylenchida. Tylenchida are thought to have evolved from rhabditide forms, the holosphere being fusion of sclerotized rhabdions. The Tylenchid sphere has a narrow lumen with ventral aperture close to its tip, and because it is formed in situ from the wall of the stoma, it is called as stomatostylate. All Tylenchids have 3 pharyngeal glands, one dorsal and 2 subventral. Plant parasitic nematodes are often classified according to their trophic niches. They may be either above ground feeders or root feeders. Now, the above ground feeders can be divided into 3 groups: bulb, stem, nematodes. Although these nematodes are sometimes found in the soil, they migrate up the stems of the plants and enter bulb, stem and foliage. The most common of the stem nematodes are species from the genus Dictylynchus. Dictylynchus dipsiki is a known migratory endoparasite which feeds on parenchymatous tissues in stem and bulbs of onion, alfalfa, clover, etc. Feeding often causes swelling and distortion as a result of the breakdown of the middle lamella of cell walls. Certain juvenile stages exhibit the phenomenon called anabiosis, which means a temporary state of suspended animation or greatly reduced metabolism, and in this way juveniles can survive in a desiccated form for many years. This often causes them to be spread from one place to another in plant material. The pine wood nematode is vectored by Cyrambicid beetle from diseased to healthy pine trees. It causes pine wilt disease, which has become a major problem in native pine forests in Japan, southern China and the USA. Depending on the age and physiological conditions, heavily infested trees may die within a few weeks to a year after infection. Similarly, Anguina triticale is a parasite of cereals, causing leaf, stem and seed galls. Dry seed galls contain quiescent juveniles. The nematodes revive with moisture and infect developing shoots or move up the plant. When flowers primordia form, they enter this tissue and stimulate development of galls instead of normal seed. According to their parasitic strategy, they can be classified into different categories. First, migratory ectoparasites, which stay vermiform throughout their life cycle and feed for a short time at several places along the route. Several genera from Tylenchida belong to this group. Tylenchorhynchus possess a short stylet and feeds on epidermis cells. causing little damage to the plant roots. No galls are induced. Second one, sedentary ectoparasites. Sedentary ectoparasites such as Paratilinchus have been observed feeding on the same cell for many days. Depending on the species, the food cell can be an epidermal or cortical cell. Third one, the migratory ectoendoparasites. Migratory ectoendoparasites, also called as semi-endoparasites, can feed ectoparasitically on roots, but very often they partially invade the roots to feed on cortical or outer stellar cells. They remain vermiform throughout the development. Examples are Hoplolaemus and Heliocotylincus, often parasitizing the grasses. Heliocotylincus species may invade roots completely, where they feed from a single cell for several days with alternating periods of salivation and ingestion. The feeding cell is surrounded by a few metabolically active cells with dense cytoplasm but without nuclear enlargement. Feeding tubes are detected in the feeding cells. Next one is migratory endoparasites. These parasites are found in the subfamily of Pratylinchinae, with Pratylinchus and Rhadophilus being the most economically important genera. Every life stage of Pratylinchus species can enter or leave the root. They mainly feed on cortical cells. Many cells are killed during their intracellular migration, and this makes the plant susceptible to invasion by other organisms. The next category is the sedentary endoparasites, which have evolved the most. They are the most advanced and specialized strategies of root parasitism. They induce specific permanent feeding by feeding cells or feeding structures from which they derive their food throughout development and reproduction. The invasive development stages are vermiform, but depending on genera, it can be freshly hatched second stage juveniles, such as in Globodera, or adult females such as in Tylincus. Soon after feeding cell induction, the nematodes become sedentary due to a degeneration of somatic musculature. The female is always sedentary. Male juveniles stay or become vermiform before maturity again and regain their mobility. In this schematic representation of the feeding sites of migratory root parasitic nematodes that we have seen, the number 4 to 7 represent Tylincid nematodes. Of these, the figure 4 represents feeding site of migratory ectoparasite Tylincorhynchus dubius. Fifth one represents the sedentary ectoparasite Criconymala xenoplax. Sixth one represents the feeding site of the migratory ectoendoparasite Heliocotylincus species, and the seventh one represents the feeding site of migratory endoparasite Pratylinchus species. To summarize, plants regularly encounter plant parasitic nematodes because of their high abundance and diversity. More than 4,100 species of plant parasitic nematodes have been described. plant parasitic nematodes are largely root feeders and collectively cause annual crop damage of more than 80 billion US dollars per year. Molecular evolutionary framework for the nematoda reveals 5 major clades, all of which including parasitic species. It demonstrates that animal parasitism arose independently at least 4 times and plant parasitism at least 3 times. Almost all plant parasitic nematodes possess a hollow, protusible, hypodermic needle-like axial sphere which serves as the interface between the plant and the nematode. It can be used to penetrate the plant cell wall or to withdraw the nutrients from the cytoplasm as well as to release the secretions. Plant parasitic nematodes occur in 3 widely separated orders: Triploncida, Dorylamida and Tylencida, with the majority being in the latter. Despite some similarities between these taxa, it is evident that evolution to plant parasitism occurred convergently in the 3 orders. And last but not the least, plant parasitic nematodes can be functionally classified according to their mode of action into above ground feeders and root feeders. Within the root feeders, migratory ectoparasites, sedentary ectoparasites, migratory ectoendoparasites, migratory endoparasites and sedentary endoparasites can be distinguished. Hope you enjoyed the lecture, and for more information, please refer to the following links. Thank you.